Here's what we'll cover today. First, we'll define what antibiotics are. Then we'll look at antibiotic susceptibility testing, why we do it, and how. We'll go over three main methods: disc diffusion, e-test, and bruise dilution. Finally, we'll interpret the results of each method. An antibiotic is a substance that inhibits or kills bacteria. It's a type of antimicrobial used to treat bacterial infections. Antibiotics can be classified based on their mechanism of action, such as inhibiting cell wall synthesis, protein synthesis, DNA replication, or disrupting membrane function. Antibiotic susceptibility testing (AST) is used to determine which antibiotics are effective against a particular bacterial strain. One of the key concepts here is MIC, the minimum inhibitory concentration. This is the lowest concentration of an antibiotic that inhibits visible bacterial growth. Common MIC testing methods include bruise dilution, e-test, and disc diffusion. Let's start with disc diffusion and the e-test. First, prepare a bacterial suspension at 10 to the 8 CFU per milliliter. Spread the suspension evenly over a Mueller-Hinton agar plate with a swab. Let the plate dry for 5 to 15 minutes. Then, using sterile forceps, place the antibiotic disc or e-test strips onto the agar. Make sure the scale on the e-strip is visible for later reading. Incubate the plate at 37 degrees Celsius for 24 hours. In the bruise dilution method, start by dispensing 100 microliter of bruise into well 1 to 11 and 200 microliter into well 12. Add 100 microliter of the antibiotic into the well of the first column. Then perform a 2-fold serial dilution up to well 10. Next, add 100 microliter of bacterial suspensions to each well from 1 to 11. Well 11 serves as a positive control with bacteria but no antibiotic, and well 12 is the negative control with bruise only. These are the results. After incubation, check for zones of inhibition around the antibiotic disc. The larger the clear zone, the more effective the antibiotic. Compare the diameter of the zones with standard interpretation charts to classify the strain as susceptible, intermediate, or resistant. For the e-test, look for a zone of inhibition where the bacterial growth stops. Read the MIC value directly from the strip where the border of the inhibition zone intersects the scale. This provides a precise MIC value for an antibiotic. Bruise dilution. Examine the wells for bacterial growth. The first well without visible turbidity indicates the MIC. Compare with controls to validate your results. The positive control should show growth, and the negative control should remain clear. Today, we'll learn antibiotics and how to do antibiotic susceptibility testing, disc diffusion, e-test, and bruise dilution. Thank you for your attention.